Hi, I'm Frasini and I'm a PhD student in Dr. Zoe Waller's group at the School of Pharmacy of University College London. In this video, we'll present our recent research paper in a simple way. Our paper is entitled QN-302 demonstrates opposing effects between i-motif and G-quadruplex DNA structures in the promoter of the S100P gene. The paper has just been published in the Journal of Organic and Biomolecular Chemistry, and you can find the link to the paper in the description box below. So let's have a look at what the paper is based on. In our group, we work on DNA, the molecule of life. DNA provides all the information and specifically the genetic code for all forms of life on Earth. In our bodies, this genetic information is held in the form of DNA, which is stored in the nucleus of our cells. DNA is made up of 4 bases: A, or adenine, T, or thymine, C, or cytosine, and G, or guanine. These bases are combined together to form our genetic code, and they include all the information needed for all the functions in our body, including normal functions, but also abnormal functions related to disease. Every region of DNA, or every set of bases that encodes for a function, is called a gene. The way that a gene is read is affected by the DNA sequence, or the arrangement of the bases. But we now know that the DNA shape can also affect the way that a gene is read, and perhaps switch it on or off. Therefore, the shape of DNA can control biological functions. The most popular DNA shape, discovered in 1953, is the double helix, also known as Watson and Crick double helix, and it looks a bit like a twisted ladder. In our group, we're interested in special DNA shapes called i-motifs and G-quadruplexes. I-motifs are formed mostly by the C base and G-quadruplexes by the G base. In this paper, we're interested in a specific gene called S100P because it is highly expressed in most patients with pancreatic cancer. An important region of that gene, called the promoter, contains C and G bases and forms the special DNA shapes, i-motifs and G-quadruplexes. For many years, Professor Stephen Needle and his group at the UCL School of Pharmacy were making many molecules that interact with G-quadruplex DNA structures and that could be potential drugs. One of those molecules, known as QN-302, was a very successful G-quadruplex binder and is currently in clinical trials for pancreatic cancer. In this paper, we investigate a potential mechanism of action of this experimental drug involving alternative DNA structures, not only G-quadruplexes, but also i-motifs. In general, we can observe the shape of DNA using light. Based on its interaction with light, we get a characteristic signature graph, and then we can determine the type of structure that we have in our samples. So this is what we did at first to characterize our S100P G-quadruplex and i-motif forming DNA sequences. We then melted those DNA structures and recorded their interaction with light during melting. This experiment allowed us to determine their melting temperature, which is a measure of how strong or stable these alternative DNA conformations are. The higher the melting temperature, the more stable the structure is. After that, we repeated the same experiment, but this time we added our molecule of interest, QN-302, in the G-quadruplex and i-motif samples. We then melted the samples and we determined the melting temperature again. What we found to be very interesting during this experiment was that the melting temperature of our G-quadruplex was increased. However, in the case of the i-motif, the melting temperature was decreased. This set of experiments showed how the QN-302 molecule affects the stability of our G-quadruplex or i-motifs at the S100P gene. More specifically, we found that QN-302 unfolds the i-motif while stabilizing the G-quadruplex. Therefore, QN-302 exhibits exceptional opposing combined synergistic effect on the i-motif and G-quadruplex at the S100P gene. It can enhance the formation of the G-quadruplex whilst unfolding the i-motif. It controls the shape of DNA in the S100P gene and therefore alters its function. This is possibly the mechanism by which this now experimental drug acts to cause its exceptional therapeutic effect against pancreatic cancer. Thank you for watching. If you have any questions, please feel free to tweet us or drop us an email. Take care and bye for now.